Okay, bone is a little different from our other connective tissues. Here we have a more organized kind of structure in our tissue. I'm going to start by putting in some layers of bone matrix to the outside of the bone. It's kind of exaggerated maybe, but anyway. This would be layers that lie right on the very surface of the bone, and then beneath that we'd have these circular patterns of matrix, bone matrix. Kind of a bullseye shaped pattern with a large opening right in the middle here. I'll get to that here in a minute. There's another large opening down here, and around that we're going to have layers of matrix. And then we get to a point where we kind of squeeze layers of matrix in between these circles. And somewhere out here there would be another one of these circular patterns. They would be tightly packed, all those circular patterns, in the bone. So each layer that I have drawn in here, the white space between the dark lines, represents the hard material of bone matrix. It's mostly calcium and phosphate compounds that come together, what they sometimes call bone salts, and the primary component of the bone salt material is what they call hydroxyapatite. But that's that calcium phosphate compound that we're talking about there. These layers of bone matrix on the surface of the bone out here are called circumferential lamellae. Lamella is just a fancy word for layer, but anyway, lamellae, many layers of circumferential matrix. Then in here we have what's called a Haversian system. This bullseye arrangement is called a Haversian system, and the layers of matrix associated with the Haversian system in here, these are called concentric lamellae. We've got concentric circles around the same center. And then those bits of lamellae, bone matrix material laid down, filling the gaps between these circular shapes, that would be called an interstitial lamella. I'll do two, so we get lamellae again. Interstitial lamellae. So you have all three forms in what we call compact bone. In a minute we're going to do another drawing with spongy bone, but for now we're just looking at compact bone, the most dense kind of bone found to the outside of most of your larger bones. Compact bone. So this is an example of compact bone here. Inside the middle of that Haversian canal is a space with blood vessels in there. I hate to use red for my blood vessels, but we'll try to do something with that. So this represents a blood vessel here, this represents a blood vessel here. So the blood vascular supply to the compact bone runs through the center of the Haversian canal in here. So I want to label that Haversian canal, that space right there in the center. And the whole thing here, this bullseye shaped system right here, it's called a Haversian system. Sometimes it's called an osteon, O-S-T-E-O-N, osteon, Haversian system or osteon. So that kind of explains the overall structure, but where are the bone cells, you probably wonder. Well, between these layers of bone matrix material here, I have a little line, and that little line represents the border of a layer of bone growth. Need to make better spaces in here. These spaces I'm drawing now are lacunae, in the same way that when we did the cartilage we had cells in lacunae. Here again we have cells in lacunae. And I'll just draw some of them. I should draw more, but just in the interest of time. Here we'll draw just a few of these lacunae or spaces in the bone matrix. So in each of those spaces we would have a bone cell, an osteocyte. Here's an osteocyte, here, here. They're really very small cells with a very small nucleus. We'll put nuclei in them in a minute here. And as I said, there should be several more in my picture. They're spaced more regularly, more evenly than what I have drawn. So those are osteocytes inside lacunae. There's a single osteocyte in a lacuna. Now, bone, compact bone, is really, really dense material, and if there were a cell out there surrounded by only dense material, it would eventually not be able to get nutrients, accumulate waste, it would eventually die. So we don't want them to die. So there are small channels that join between one osteocyte and another, all the way down to the middle where the blood supply is. And these tiny little channels that go through the lamellae, through the layers, are called canaliculi. That just means tiny canals. So these tiny little lines, these little canals, connect all through the compact bone. They're not in the eversion canal there. Going through all the lamellae, joining the bone cells to neighboring bone cells. So those would be all through here. All these canaliculi. And I don't know where to label that. Let's just put it right here. Canaliculus. A single small canaliculus. So that's compact bone, from the outside of the bone moving inward. On the very outside of the bone, when we talk about bone later, we'll talk about the periosteum, and there's some bone forming cells on the outside that are laying down the bone material on the outside to make more layers where it's needed. But anyway, this is compact bone. It has the flat layers of matrix on the outside, and then we get into those eversion systems around the blood supply to the compact bone that's just under the surface of the bone. And usually in most bone, there's just a fairly thin layer of compact bone before you get to that spongy type of bone on the inside of the bone toward the middle. In your thickest, heaviest femur bone, there might be almost, well, not a half inch, but at least over a quarter of an inch, maybe 3/8 of an inch of compact bone making up the wall of some of that, some of those bones in there. So that's about the thickest. In most of your bones, like your finger bones, your bones of your skull and so forth, it's really, really thin, that compact bone layer on the outside. So this is compact bone. Next we're going to do spongy bone drawing. Okay, I'm drawing spongy bone. We've just put in kind of a spongy layer in here. I'm going to put a red space in there where the bone is not, where there's no bone matrix. So we're just going to fill this in. This space right here, these cavities in the spongy bone, this is where you produce your blood cells. This is where you have all the different kinds of blood cells your bones produce. That happens primarily in these small spaces in the spongy bone. And these are all kind of connected up to one another and eventually to the bloodstream so that we can disperse those blood cells into the blood and get them out into circulation. Imagine that over here on this side we have the layers of compact bone, the haversian systems and all that going on out here, that compact bone arrangement to the outside, and then as you're coming into the bone toward the middle, toward the marrow cavity in the center, you're going to encounter this spongy bone structure. So each of these little connecting lines right here can be referred to as a bone spicule or a trabecula. Bone spicule or a trabecula, this is spongy bone. Now the bone that makes up the component in the spongy bone, that bone matrix material, is a little different than what we saw in the other. It's just, if you take a cross section of a single bone spicule, it'll look something like this. That's not a haversian canal in the middle. There's cells all around here, osteocytes around here, and let's go ahead and draw those in before I draw in the canaliculi. So anyway, in here you'd have all those little connecting lines all the way down to this, toward the middle of the bone, down in here. So you have your canaliculi coming in. So your nutrient supply is from the outside, there's no blood supply in the middle of one of these bone spicules that runs through here. So anyway, there's a network of bone spicules all connected to one another, and sometimes this completely fills the middle of the bone. So if you looked at one of the plates of your skull, between the outer compact bone and the inner compact bone, you would have a layer through there that's all this spongy bone. If you cut through the humerus in your arm, you'd have the outer compact bone, a very thick compact bone. Then you'd have a little bit of spongy bone, and then you'd have the marrow cavity to the inside of the bone where there'd be a lot of fat. And there would be some more blood-forming tissue, but there'd be a lot of fat also on the inside of that structure. But this is spongy bone made of bone spicules or trabeculae, and that's what it would look like if you cut one in cross-section. Now we're going to talk about blood for just a minute. The matrix material of the blood is what we often refer to as blood plasma, the fluid component. And then the remainder of the blood we refer to as formed elements. Now, generally we think most of the types of formed elements we have in the plasma, they're going to be different kinds of blood cells. So why don't we write just blood cells here? Our platelets are not actually full cells. The platelets that are found in the blood are fragments of cells. So we could see here, instead of formed elements, blood cells and blood cell fragments, or just blood cells and thrombocytes or platelets. But anyway, so those are two things make up our blood. Within our formed elements, we have different categories of blood cells. First of all, we have our red blood cells, also known as erythrocytes. The red blood cells. Then we have our white blood cells. known as leukocytes. Among our white blood cells, we have what are called granular leukocytes. That includes neutrophils, eosinophils, and basophils. We're going to make drawings of all the blood cell types here in a minute. We're just trying to get a list on the board here. So if we have some granular leukocytes, it stands to reason we would have some agranular leukocytes. And so that's, I'll move the camera a bit. Two types of our white blood cells that are agranular leukocytes. They are the lymphocytes, and there are actually several kinds of lymphocytes, but in the visual overall appearance, they mostly look the same, the lymphocytes in the blood. And then there are the monocytes. Lymphocytes and monocytes. So we have red blood cells, then we have white blood cells that include granular and agranular types of cells. And then we have the thrombocytes or platelets. That are vital for blood clotting. And like I said, they are a fragment of a larger cell type that's found in the bone marrow called the megakaryocyte. So next, we're going to start drawing these and looking at the different examples. Okay, I've got a blank board here. I'm going to try to fill it up with drawings of blood cell types. Give you some feel for what that looks like. It's good to make the drawings, put your pencil to paper, and construct these illustrations yourself just to help you remember the differences. So first of all, a red blood cell, we'll start with a red marker, I guess. Red blood cells are, if you look at them in one direction, they're a nice disc shape. If you were to look at it on side, it would have this kind of profile to it. And if you had it kind of at an angle, you would see that it's concave in the center. Kind of like a jelly-filled donut or something in there. So anyway, or a Certs candy mint. So that's the overall shape of a red blood cell. So it has this kind of concave region in the center. Red blood cells, or erythrocytes. They are smaller than your other blood cell types except for thrombocytes. So the next cell I'm going to draw here, we're going to do a series of granular leukocytes next. Need to make it a little bigger than a red blood cell. And inside of a neutrophil, there's a nucleus that has an odd shape. You'll often hear the term polymorphonuclear leukocyte applied to this. That just means they take many shapes, but there's usually 3 or 4 or more lobes to the nucleus that are kind of strung through the cell like this. It's a fairly large cell, and it's a neutrophil. It's a granular cell. That means there's granules inside the cytoplasm, and they don't stain particularly well in a neutrophil, so it's a little hard to see the granules. I'm just going to use my brown marker here and give some light stippling to it and remind us that it's granular, but the granules are hard to see inside of a neutrophil. So that's my neutrophil cell. Next, I'll do my eosinophil cell. It's about the same diameter, same size. I've drawn it here a little bit bigger, didn't mean to, but that'll do. Its nucleus is not quite as strung out typically as the typical neutrophil. It usually has 2 lobes, could have 3, but anyway. This is typical eosinophil nucleus I've drawn here. Now, eosin is a kind of a stain, an acid stain that is used to help visualize features of tissue under the microscope. And the granules within an eosinophil stain with that red stain, that eosin stain. So the cytoplasm filled with granules, small staining granules, and here the eosin stain adheres to all those granules. So that's an eosinophil. Next, a basophil. Basophil also in the same group, a granular leukocyte. It has a divided up nucleus like we have on these others. But you can't, well, often you can't see the shape of the nucleus inside there because of the dark staining granules. This is called a basophil because the basic stain, instead of the acid stain like we had with the eosinophil, the basic stain will adhere to these granules in this granular leukocyte. And the granules stain these in the same color as the nucleus. It's common for some of these dark staining granules to be right out on the surface of the cell, so it looks almost bumpy in places around the margin of the basophil. So there's neutrophil, eosinophil, and basophil. They have granules on the inside. These don't take up either the acid stain or the basic stain. The eosinophil takes up the acidic stain, and the basophil takes up the basic stain. So those are our granular leukocytes. Now for a couple of agranular leukocytes. The lymphocyte, usually smaller than a neutrophil but bigger than a red blood cell. And they have a nucleus inside that nearly takes up the entire space of the cell. So I often tell students to help them recognize this in the microscope, that they're looking for a cell with a nucleus that fills the cell, and that nucleus is about the size of a red blood cell. Nice big round nucleus. It's fairly easy to recognize. It's not like any of the other cell types. So sometimes all the blue in the basophil kind of looks a little bit like a solid blue that we see in the nucleus of a lymphocyte, but usually in a lymphocyte there is a edge, at least some space around the margin between the nucleus and the membrane that you can see in there. So that's a lymphocyte. Next, the monocyte. The monocyte is our largest of the the white blood cell types. So here's a monocyte. Its nucleus is usually shaped, I sometimes say it's like a bent pillow or more like a bean shape or something like that. Has a very large nucleus, but it usually has an indentation on one side. So there's our large monocyte. So we've seen red blood cells, we've seen two kinds of white blood cells, the granular leukocytes and the agranular leukocytes. The platelets, I've mentioned before that they are actually fragments of cells. They are not cells of themselves. So these things I'm drawing in here now are platelets, cell fragments in the blood, part of your formed elements of the blood. Now, I've drawn this picture just to help you see what they look like. If you were to see them under the microscope, they would take on generally these kinds of appearances, but in terms of numbers, most of the blood is made of red blood cells, the erythrocytes. So there'd be thousands and thousands of these on a blood slide. You can look around and find a fair number of neutrophils. Those are fairly abundant, as are lymphocytes. Those are very common cell types in most blood samples. Your monocyte would come in next, the next most common blood cell type. But the eosinophils and even more so the basophils are rare blood cell types when you're just looking at a blood slide. You have to look longer and search more to find a single erythrocyte than you would these other cell types, and even more so, like I said, for the basophil. They're even less common than the erythrocyte. The basophil is less common than the eosinophil. So those are your blood cell types.